Hi all, I'm Thanuja Fernando, conducting a PhD in Bioinformatics at the University of Guelph. Today I am presenting my research, testing bioinformatics approaches for building large phylogenetic trees by placing DNA barcode sequences. First, I'll walk through by describing the biological problems that leads to conduct my research study. As we all know, biodiversity is the foundation of ecosystem services such as pollination and pest control, which is essential to sustain agriculture and humanity. So, maintenance of biodiversity is vital for ecological stability and sustainable production of food and agricultural products influencing food security, nutrition, and livelihoods. Nowadays, most of the farmers struggling to control invasive species like weeds, pests, and pathogens that can harm their crop production and farmed animals. But with the knowledge of biodiversity in a given system, it's possible to improve resistance to invasive species and resilience of that system. According to recent studies, 98% of all species are yet to be published in reference databases like NCBI and BIOLD. That means almost all the data are missing from reference databases. Therefore, there should be an approach for taxonomic and functional annotation of novel DNA sequences, even in the absence of species-level match in databases. DNA barcoding, phylogenomic, and multigene are some approaches for phylogenetic placement and for phylogenetic rebuilding. As these approaches have strengths as well as weaknesses, we have a great chance to combine these approaches together. So for that, first we need to generate a phylogenetic tree using multigene sequences. And then I place DNA barcoding sequences which represent by these levels on the generated backbone tree in order to find the optimal position for new query species. After a thorough study, I was able to determine the gaps in knowledge related to this study. So the first one is still there is no large-scale comparison among phylogenetic software tools and methods in terms of phylogenetic accuracy and computational efficiency. The second one is still researchers are yet to fill the gaps on how much data is needed in backbone tree in order to generate a reliable final tree. Likewise, it has not yet been studied on how the phylogenetic pattern of species coverage in the backbone tree influences phylogenetic accuracy. Then the fourth one is still there's no third-party benchmarking for phylogenetic placement. And finally, there's an incomplete understanding on performance of phylogenetic placement software tools with Core 1 animal barcoding data. The goal of this research is to develop combined approach of multigene and placing DNA barcode reads on backbone trees to determine how novel sequences fit into the evolutionary context. In this study, I have used History of Life dataset as it consists of 27 genes from several sources like Bold, Rabowski, and Neal. Moving on to detailed workflow. First, I have generated 100% complete backbone tree using full multigene History of Life dataset and kept this tree aside for downstream analysis. Then, I've built different level backbone trees with random samples at 80%, 60%, 40%, and 20%. As the next step, I've added the missing species back into each level backbone tree based on Core 1 data. And finally, I've plotted the completeness of the backbone tree against several metrics of phylogenetic concordance and topological distance. There, I've calculated the phylogenetic concordance by comparing matrix generated using each sampling level's backbone tree to the 100% reference tree. Here, I've repeated the same procedure 10 times in order to obtain replicates of different levels of sampling completeness in the backbone trees. Moreover, I've used multiple metrics in this project to explore different components of phylogenetic accuracy. Robinson-Fold metric and generalized Robinson-Fold metric come under topology-based methods, while congruence among distance matrices come under distance-based methods. Then I've analyzed the results for each metric using a statistical test. Here I compared the measures of phylogenetic accuracy, the response variable, across the levels of sampling completeness, which is the predictor variable, using one-way repeated measures ANOVA. The reason for the selection of repeated measures approach is the sequential data dropping technique, because the species included in each level of sampling completeness were not random for each resampling run. Also, I tested whether my data conform to the assumptions of this test. I've used RStatics package and visualized the data using violin plot methods to check outliers. Then I've checked the variances of differences between groups with the March least test. If the variances are not equal, then I had to check the two different types of corrections, Greenhouse Geyser and Hunt-Felt correction. After that, I've conducted Shapiro-Wilk normality test and visual inspection using QQ-plot to check whether the dataset is normally distributed. Finally, I applied the statistical technique called repeated measures correlation to determine the overall within individual association among paired measures. Okay, so far I've described the methodology, and here onwards I'm presenting the results of the statistical test. According to the values of repeated measures ANOVA, Robinson-Fold metric and path distance metric have statistically significant difference in phylogenetic accuracy at each level backbone tree, and congruence among distance matrices has no significant difference. Then, results of the first assumption show no outliers in this dataset. To prove that, I had to use violin plots. Here you can see three violin plots. The first one is for Robinson-Fold metric, the top one is for path distance, and the bottom one is for congruence among distance matrices. In each graph, the horizontal line represents the backbones at different levels, 20%, 40%, 60% and 80%, while the vertical axis represents the phylogenetic accuracy. By looking at the nicely distributed points, we can prove that there are no extreme outliers in this dataset. The values of the second assumption show a normal distribution in my dataset, as all the p-values are greater than 0.05. As my dataset is so large, I had to use QQ plots to check whether these results are correct. The reason for using QQ plot is, at larger sample sizes, the Shapiro-Wilk test becomes very sensitive even to a minor deviation from normality. Here, the x-axis shows the theoretical normal distribution, while y-axis represents the sample dataset values. Also, you can see that there are separated plots for each level sample completeness at 20%, 40%, 60%, and 80%. As all the points fall approximately along the reference line, we can assume normality. As the p-values in final assumption are greater than 0.001, they represent the variances of group differences are equal. Further, the results of the repeated measures correlation imply a strong correlation within individual groups of Robinson-Fold metric and path distance, while congruence among distance matrices has no correlation. Then I've used graphical representations of metrics to show the linkages among points at each varying levels of sampling completeness. By looking at the trends of the plots with different level backbone trees, which means the sampling completeness on the x-axis and phylogenetic accuracy on the y-axis, we can compare the difference between topology-based metrics and distance-based methods. So, the plots are Robinson-Fold metric, path distance, shared phylogenetic information, mutual clustering information, Nye similarity, matching split information, Jacquard-Robinson folds, and congruence among distance matrices. By considering all the outcomes, we can conclude that topological accuracy strongly relates to the backbone completeness, while distance-based methods have no significant difference in accuracy when the sampling completeness is increasing. So, if the purpose of the future research is taxonomic revision, then topological accuracy is important as the accuracy is strongly related to the sampling completeness. But if the purpose of the future research is finding a large-scale tree properties and diversity, then distance matrices are important as there is no significant difference in accuracy when the completeness of sampling increases. Ultimately, this work will help other researchers to build large trees to study phylogenetic diversity and predicting species traits in agricultural biomonitoring. In addition, this study will contribute to early detection of potential pest and other non-native species while capitalizing upon the large data set being generated from understudied habitats and regions of the world. So, these are the references, and thank you so much.